Hello everyone, I am Pratibha Tripathi, postdoctoral fellow in Partha Das lab at Monash University. Today I am going to talk to you all about how we can break boundaries for PWS research using brain organoid system for advancing therapies. So to begin with, I would like to give you a brief introduction about Prader-Willi syndrome. Prader-Willi syndrome affects 1 in 15,000 people irrespective of their race and gender. It is a genetic disorder that affects appetite, growth, and cognitive function of an individual. So how we can benefit from human brain organoids for PWS research? Human brain organoids closely mimic structure and function of the human brain. They facilitate to study intricate brain development in a controlled lab environment. Also, they avoid any ethical concerns associated with studying live human brains. So what experimental design required to study PWS using human brain organoids? In order to generate organoids from PWS individuals, firstly we need to generate stem cells from healthy and PWS individuals. This is an easy process. We can get pluripotent stem cells from any individual using 10 ml of their blood. Using these individual pluripotent stem cells, we can generate brain organoid or so-called mini brains, which include all the cell types present in the human brain. Next, we can characterize these organoids in terms of their cellular, genetic, and functional composition. Here, I am showing you an example of organoids generated in our lab and has various cellular compositions and express different markers which mimic early developing brain. In this video, you can see how different cell types are generated within the organoids and they are communicating with each other. Next, using brain organoid, what we can expect in terms of their outcomes for the PWS research. First, we can look at the gene expression changes from the organoids generated from healthy and PWS patients, and how various genes, mainly in terms of appetite regulation or brain development, are deregulated. Next, we can look at the neural network within the organoids and how they are impaired in PWS derived organoids and how their synaptic functions are looking. Also, within the organoids, functionally we can look at that how they respond to various neurotransmitters and hormones. And lastly, you can even compare these organoids to post-mortem human brain tissues. And this can allow us to look at any shared molecular and cellular abnormalities existing within these organoids, which can actually correlate to the post-mortem tissues derived from PWS individuals. So as we can see, human brain organoid can serve as very powerful tool for understanding diseases like PWS at molecular, cellular, even at functional levels. So I would like to let you all know that we in our lab have successfully generated organoids from PWS individuals, and this can actually highlight its as a potential tool for various therapies for PWS. We can use these organoids generated in our lab for drug screening and testing various pharmacological interventions. We can also use these organoids for precision medicine, which is a very patient-centric approach where treatments can be customized based on individuals' genetic profile and disease characteristics. This is a cutting-edge technology that has potential to improve patients' outcome and minimize adverse effect and ultimately lead to efficient healthcare. And finally, these organoids can be used for also gene therapy. With this, I would like to acknowledge our lab, mainly Dr. Partha Das and Monash University, where I did all this work. And lastly, I want to really thank Prader-Willi Research Foundation Australia, which provided us the funding to do this kind of research. And thank you all.